So welcome back. This is part two of our lecture on viruses, COVID-19, and vaccines. Part two will be featuring specifically COVID-19 and vaccines. So let's talk about COVID-19. First of all, we're all living through this pandemic right now, and I'm sure that you have some information or a lot about it, and we all have our memories of when the lockdowns occurred and all that in 2020. That was March, I believe. Here are some current stats as of at least January 12th. There have been more than 600 million cases. Probably the reality is many more, as not all cases are recorded. These are just official cases, so around 7 million deaths. Again, probably the actual numbers are much higher. For Canada, these are the official cases, so 4.5 million cases and about 50,000 deaths. Again, these numbers are much, much higher. This past year, most cases have not been recorded. People have just been staying at home. There have been many undiagnosed cases and so on, but those are the official numbers. So this has been something to live through, and thankfully this disease has not been as virulent as other pandemics, such as the 1918 flu, but it has obviously changed our lives and our economies and everything. So lots to think about, and let's talk about the biology of this thing. Before I get into that, I just want to say that a lot of people seem to be shocked. They watch these science fiction movies and these pandemics and think that they're rare. It turns out that outbreaks, epidemics are actually a very normal thing. Here's a few that I can recall from my lifetime, and there are many, many, many more. And you can see that some of them have directly impacted us in Canada. So West Nile virus is something that arrived in Canada in the year 2000. And some of us have probably had it. It's been a bit more of a mild disease, a bit more of a risk if you're an older person. The first SARS arrived in Canada and only in Toronto in Canada. And this was in the years 2002 and 3. And this was a very serious disease. You can see the ones that have double stars beside. These are diseases that are killing a high number of people. So thankfully that did not spread beyond Toronto. It was actually easy to detect and isolate if you're looking for that kind of information. Notice H1N1. There's another pandemic we've all lived through that has arrived in Canada. So this is a regular thing. In your lifetime, there is usually some sort of epidemic at least every 5 to 10 years, and many of them we don't even notice, right? Zika, again, not relevant to Canada because it's spread by tropical mosquitoes. But the big one that has affected all of us, of course, has been this new one, this COVID-19 pandemic. And this is what we want to talk about the biology of the virus and a little bit about vaccines. So first thing, of course, is where do coronaviruses come from? So before the year 2000, there were 4 coronaviruses that were known to humans that infected humans. And all of these probably crossed into humans hundreds of years ago, if not more. And they have funny names. You can see HCOV and L63 and so on. And most of them, we believe, have come from bats or rats or rodents. And usually there's some sort of intermediate animal that we can track its origins to, because we're not necessarily living with bats all the time, but we might be living with our livestock. All of these are low pathogenicity, meaning they don't make you very sick. In fact, they cause the common cold, and we get these infections quite regularly. Every 6 months to every couple of years, we get infected by these and usually don't think too much about it. So coronaviruses up until the year 2000 were not thought of as serious infections. So what happened in the 2000s? 2 new coronaviruses popped up. The first SARS, we're calling it SARS Classic now. That was very scary. It hit a few places like Hong Kong and South Korea and Toronto. So that was something that if you know anyone living through that at the time, ask them about it. It's kind of some interesting stories. There's another one, MERS coronavirus, means Middle East Respiratory Syndrome, I believe is what the acronym stands for, and it's associated with camels. And so places in the world that have camels, so the Middle East, and very, very serious disease. And still there are outbreaks every few years on that. So we are starting to take coronaviruses a little more seriously, and unfortunately a new one arose. So SARS coronavirus 2, and this one now is considered a human disease, and we think it came from bats. Notice there's a big question mark about what the intermediate might be. We don't really know. There are some guesses. A lot of people thought it was a pangolin. Now you're probably thinking, what is a pangolin? Well, it's worth it to look that one up. In fact, I have some nice pictures for you. That is a pangolin. So it is an animal and it has scales on its skin. And this is found in various parts of Asia and Africa, I believe. I don't know if they're found in other parts of the world. And there are a few different species. And they're very cute. They like to roll up in, they carry their young. You can see there's a baby one. And they roll up in little balls to protect themselves. And this was at one point believed to be the intermediate host, but I don't think we actually know. And it's very hard to track that kind of information because viruses are mutating and changing all the time, unfortunately. So here is the virus. So SARS-CoV-2 stands for Severe Acute Respiratory Syndrome. So severe means bad, acute means quick, respiratory means you're breathing, and syndrome, of course, means that's describing the disease. And it's a coronavirus. So corona means crown. And the person who discovered this was actually a Canadian scientist, a Canadian female scientist in the 1960s, and she thought it looked like a crown. So she actually named the virus coronavirus. So this one is SARS-CoV-2 because it's the second in its group to be found after the original SARS in the early 2000s. So notice it has your typical virus structure. It has an RNA genome on the inside. There is a capsid. They're not really showing it very well. And on the outside is this glycoprotein, the spike protein. So that's something we're going to talk a little bit about as important to infection, but also important for immunity. Here is the genome. So how many genes? Does it say how many genes that has? It does not. Apparently about 30,000 base pairs and a handful of genes, some that we're very familiar with from other viruses, the spike protein being the one that is going to be involved in infection. So you can see I have it highlighted there. There's the spike protein right there. Or it was there. There's the spike protein. So before we get any further, I just want to address what is a variant? This is a term we've been using to talk about the new versions of this virus. And so just some quick definitions. First of all, what is a mutation? A mutation is the change in the genome sequence. So the change in the code. So RNA has A, U, C, and G. So if one of the A's is changed to a G, then it has a mutation. And so mutations are a very common thing. They're happening in your body. They happen in viruses. They're a natural biological process. So what is a strain? A strain is a subtype. So it's where we say something is different, and sometimes it has distinct properties. So for example, there are strains of E. coli that do not make us sick. That's what most of us have in our intestine. But sometimes people go and they travel to a part of the world and they drink contaminated water and they come back with a pathogenic E. coli that gives them diarrhea. So this is a distinct property. It's giving somebody a diarrhea or not. So what is a variant? A variant is another subtype and is different from the original. So it has some mutations, but we're generally not saying it's different enough to be called a new strain. So who decides if something is a strain or a variant? I'm not sure. Probably the people that are researching it. And so at this point, we're using the word variant quite a bit. So variant is different, has some mutations, but it's not a new strain yet. So there are a whole bunch of variants. There's actually thousands that have been discovered with the coronavirus. Like I said, mutations are natural, normal things. And these variants have had a few different names, but they've been sticking with the Greek letters now. So alpha, beta, gamma, delta, omicron, etc. And so they have some different properties, or at least it seems this stuff is really hard to study, by the way. So notice, for example, that we think the variants have increased transmission. How do you study that? It's actually really hard science to do because unfortunately humans have weird behavioral things that they do, right? Sometimes we have lockdowns. Sometimes we remove the lockdowns. Sometimes there's cultural things going on in certain countries. You know, if you are living in certain parts of Asia, it's very natural and common for many people to wear masks. We're a lot more resistant here in Canada, for example. But some of the data does seem to show that we're looking at increased amounts of transmission and maybe the vaccines are slightly less effective. The vaccines still do seem to be effective, just not 95% effective. Maybe they're 70% or something like that, which is still pretty good. That's a B minus in my class if you get a 70, and that's not a bad grade to have. In terms of severity, probably less severe we're looking at, but we don't really know. Probably most of the human population now is a point where we have some immunity, either due to vaccines or having caught the disease. So it's very hard to study unless you have a lab and you're going to purposely infect a bunch of people that have never been infected before. That's the only way to really find out this information. But those are the variants. And so there's lots of reason to think about these things because, of course, we should be concerned. Are they making us sicker? Are the vaccines still working? And that's all part of the public health data that professionals need to make decisions around. Let's take a quick look at the life cycle of coronaviruses, specifically SARS-CoV-2. You can see the very first step is like the very first step of all viruses, where you have the surface of the virus binding the surface of a cell. So in this case, you have those spike proteins, and they're binding a receptor on our respiratory tract called ACE2. And it's also found in other human cells, by the way. And the genome gets inside. So this is an RNA genome, of course. And so what happens to that RNA? The RNA gets replicated by RNA polymerases, which is shown there in step 3. And some of those RNAs go on and they're found by ribosomes, shown in step 5. And the ribosomes make new virus proteins. So after that, the viruses eventually start to assemble. Notice some of the viral proteins are made in the Golgi body. That's where a lot of membrane proteins are being made. They assemble. Eventually, the viruses are released from the human cells by exocytosis and maybe going to go find new cells to infect, unfortunately. So very typical virus life cycle. You've got the binding, you've got the gene expression, you've got the release. Same 3 things you're going to see in all viruses. This virus does not need to go to the nucleus. It all happens in the cytoplasm, which is really common with RNA viruses in general. So let's talk a little bit about the disease. The disease, of course, is COVID-19, which stands for Coronavirus Disease 2019. So that's worth knowing, by the way. That was a bonus question last year on a midterm. What was COVID-19 and what was SARS-CoV-2? So worth it knowing for you. Possibly be on our midterm as well. So this disease was first described in 2019. This is why that is in the name. So hopefully everyone's kind of familiar with all the symptoms, many respiratory symptoms, cough, fever, runny nose, those kind of things. And some people, shortness of breath. In about a third of us, loss of smell and taste. That happened to me when I got COVID-19 for the first time. And it lasted about maybe 2 or 3 days where I couldn't taste things. It was a very interesting experience. So I just want to spend a minute comparing COVID-19 with some other respiratory diseases. I'm not expecting you to compare all of these on a midterm or anything, but it's kind of some interesting things to think about and a few points to make. So first of all, COVID-19 has its own virus. It's a coronavirus. Influenza, also its own virus called influenza virus. The common cold is caused by many types of viruses. I've already mentioned rhinoviruses and coronaviruses and adenoviruses. There's something like 200 or 300 different types. Notice these viruses might have different genomes, different structures. So we're looking at different viruses and therefore different diseases. Do we have vaccines? For COVID-19, yes, there are at least 4 vaccines in Canada. I think there are actually 6 now. I think I have that on another slide. Influenza, we have an annual flu shot, and for colds, there's no vaccine at all. So the other thing to look at is the symptoms. So COVID-19 is very common for people to have a fever and a cough and many other possible symptoms. If you look at influenza, often you're looking at many similar symptoms, even more common to have headaches, aches and pains, those kind of things, and a sore throat. The common cold, sore throat is more common, less common for people to have a fever, and is often more mild. So you're looking at different profile of disease symptoms because they are different viruses infecting and affecting the body in different ways. So one more thing to think about is how do viruses actually cause diseases? really two different ways, depending on how you look at it, sometimes broken into three different ways. Number one, I've mentioned that already on part A of the lecture, is that viruses will kill cells. And so when viruses kill cells, they can cause symptoms. So if the virus, like a cold virus, is infecting your nose and the back of your throat, you're probably going to have an itchy nose. Maybe the back of your throat is sore. That's the virus killing those cells. SARS-CoV-2, sometimes it gets into the lungs and kills lung cells. And so people sometimes have a hard time breathing. It kind of depends on how deep the infection gets into your body. And so that's number one. Number two way we get disease symptoms is because of our immune system. So our immune system universally, when it reacts to infections, we get things like fevers and chills and fatigue, those kind of things. And so that's very, very common for people to get while they're sick as well. You can see number three is more the long-term effects, which are also really just part of the immune system. So things like inflammation and whatnot. So sometimes people have a little damage to their lungs and have difficulty breathing for weeks and much, much longer. And again, that's part of the immune reaction to the virus, is usually cleared after about 7 to 10 days, sometimes a little longer. But people are still sick for a while because of those immune reactions that are still going on. And that's something we're trying to prevent when we treat people with severe illness. There's another picture just showing COVID-19 infection and showing the lungs, of course, and the respiratory system, which is what is going to be affected by the virus. So the last part of this, I want to talk a little bit about immunity and vaccines, because this has been a big topic of discussion around the pandemic in many, many different ways. So first of all, how does immunity work? So generally you get infected by something, which you can see at the top. We've got this virus is infecting the body, and immune cells come along and they can recognize it as something that is foreign to the body. So something that is not human. And so these immune cells, you can see there's different types. We've got these T helper cells, we have these B cells, and so on. I don't need you to worry about the different types of human immune cells. They can make something called antibodies. So these antibodies recognize this foreign invader. And so when you're infected for the first time, it takes time to develop immunity to something. So when you're infected the first time, this can take days or much longer for your body to react and mount an immune response, which ultimately uses these antibodies to destroy the infecting agent. After the first time, you have memory cells. So these memory cells will produce antibodies much quicker. And so rather than taking days, we're talking about minutes to hours. And the second time you're infected, you may not even get sick, depending on your immunity and how fast it happens. And usually in almost all cases, it's at least much less severe disease. So this is the whole idea behind vaccines. If we can somehow give us the memory of an immune response without making us sick. So that's what I want to talk about here in the last part of this course, is some different virus or vaccine strategies or vaccine types. And each of these I'm going to talk about. And the whole idea behind all of these, like I said, is to give your body immunity to something so that you're not going to get as sick and hopefully not even have disease at all. So let's talk about these. So the classical types of vaccines are these two here, attenuated or inactivated. So these technologies have been around for, I don't know, something like 400 years. And what you do is you take a virus and you either kill it. So inactivated is sometimes called a killed vaccine. So killed. Or you can weaken it. So how do we weaken something? This is the word attenuated. There are many different ways, genetically or chemically, many classical ways. I'm not going to get all the methods of making a virus weaker. But then you inject that into the human, and the human can mount an immune response against something that's killed. So that if it's killed, the virus is not going to infect you. If it's weakened, it's probably not going to cause disease or very mild type of disease. And so the flu shot is a killed influenza virus. The mumps, measles, rubella vaccines are weakened. These vaccines have been around for hundreds of years, really. Very classical kind of approaches. You can see there's the immune cell making the immune response, and so that's the whole idea. So we're not using these for coronaviruses, at least in Canada. These classical techniques have some drawbacks. They take time, and they take really secure bio safety facilities where you're growing up viruses, pathogenic viruses that could kill people. So this takes time to develop and a little bit more safety. And so these types of vaccines have been developed in other parts of the world, China and Russia, for example. But we're able to do other types more quickly here in at least the US. Canada was a little slower with vaccine development, unfortunately. Some vaccines are protein-based, meaning rather than inject the person with the entire virus, they can just take the spike protein, for example. So you can do that. You could make it up in E. coli or a plant. And so you're kind of skipping the steps where you have to grow up the actual virus. And there have been a number of companies working on this kind of thing. And this is another classical method. We're doing this with hepatitis B and tetanus shots, diphtheria shots. Those are all protein-based vaccines. So the new vaccines that we've been hearing a lot about are nucleic acid vaccines. So what does that mean? It means we're going to inject DNA or RNA into a human. So this is actually a really interesting idea. The DNA ones haven't really worked. At least we don't have any in humans. So I'm not going to talk about those, but we'll talk about the RNA ones. And so RNA, of course, is the instructions for building something. So we actually, the vaccine companies, Moderna and Pfizer, coat their RNA in some sort of lipid. They don't tell you what it is. That's a trade secret, of course, but that lipid helps deliver the RNA to your cells. And so what they do is this is injected into your cells, and it's taken up by usually it's in your shoulder, so your muscle cells take up the RNA. The RNA is found by ribosomes. The ribosomes make proteins. And what protein are we talking about? In this case, it is our spike protein. So there's our spike protein. The spike protein is released by the cell. Our immune cells find it, and they make an immune response. So this is pretty cool. Notice it says here, no licensed vaccines use this technology. This is an old image. We, of course, have Moderna and Pfizer, and they've been around for 2 years now. So this is really cool type of technology, and it's quicker to make. All you need is a genome sequence and the ability to make RNA, of which these companies have. And it's much quicker. You don't need to be growing up viruses, and much safer in many ways. The last type of vaccine I want to talk about is called a viral vector vaccine. And this is what the Johnson vaccine and the AstraZeneca vaccine, the technology they're using. It's basically a DNA-based vaccine, but the delivery mechanism is they're using a weakened or non-human virus. So AstraZeneca is actually using a chimpanzee adenovirus. So it can only infect and cause illness in chimpanzees and not humans. And this is being used to deliver DNA. And that DNA, of course, is instructions, and those instructions are going to be used to make, once again, the spike protein. And then that's going to be make an immune response. So how many vaccine types are there for COVID-19 in Canada? I was just looking this up, and it turns out there are 6 types now. You may not have heard of all of them. Not all of them have been rolled out or are being used in Alberta. But you can see there's the Pfizer vaccine right here. That's meant to be a star. And the Moderna vaccine right here. Those are messenger RNA vaccines. The AstraZeneca and the Johnson & Johnson, those are vector vaccines. They're using different types of weakened viruses. And there's a couple of new ones. I don't know too much about this plant-based one here. It's called Covafenz or something like that. And this Novavax is a new vaccine, which is just using the subunit, the protein, the spike protein. And so you can see 4 of those are now available in Alberta. Most people are getting the mRNA vaccines as part of their vaccinating protocols. So a few more things to say before we wrap this up. First thing to say is, of course, what about side effects? This is the biggest question and concern of people that are getting their shot, is how is this going to affect me? And it's a fair comment and question, because of course you want to know, is this gonna hit me hard? And here's the common side effects. Almost everyone has a little bit of pain at the injection site. And depending on your immune system, some people get headaches and fevers and chills and things like that. And these are all basically signs that your immune system is working. It's the immune system trying to have an immune response. In almost all cases, these are mild symptoms and not nearly as bad as getting the disease, and they're usually short-lived. I remember when I got my booster for Pfizer, I had the next day, I had a headache. It lasted most of the morning, and that was the end of my symptoms, as opposed to having a fever for 3 days straight. There are some other rare side effects, and these are talked about in the news quite a bit. I'm not going to get into all of these, other than to say that most of them are rare, meaning about 1 in every maybe 100,000 people get them. Some are much more severe than others. The most common one is severe allergies. Sometimes people have egg allergies, and some vaccines use eggs. So something to think about. If you're concerned about that, I'm not going to give you all the details on that. We can talk about it anytime you want. It would be a whole nother lecture in itself. But it does happen. And this is, unfortunately, like anything has side effects or risks. If you drive a car, you have a chance of dying in that car. How good are the chances? Much better than dying or getting rare side effects from a vaccine. In fact, you have a 1 in 100 chance of dying in an automobile in your lifetime. It doesn't stop us from using automobiles. The benefits outweigh the risks. Same thing with vaccines, although the risks are much, much more rare and usually not as serious as a car accident. Some last thoughts. I'm a really big believer in vaccines. 400 years of history and technology. They have not been perfect, but what is perfect? But if you look at all the diseases that we have eliminated in our lifetimes, vaccines have been one of the most amazing and impactful health medical discovery of all time. And I do worry about vaccine hesitancy. So if you want to talk about vaccines and get some information on them, I would love to talk to any of you at any time about my thoughts on these things. And I do have quite a bit of knowledge on these, so I'm somebody worth talking to about this. Here's some data of actually many diseases that probably most of you have never seen in your lifetime. So diphtheria, measles, mumps, pertussis, polio, rubella, smallpox, tetanus. Talk to somebody old about these things. Talk to them about smallpox or mumps or measles. These are things that used to be huge fears and something that everyone got as children. And now we're looking at very few cases in Canada ever, which is kind of awesome. So what about the future vaccines? So here are some things that I was reading about that are very fascinating. The big one that we'd like to see is better vaccines, or even any vaccine for malaria, tuberculosis, and HIV. Actually, there's a new malaria vaccine that just came out last year, so that's progress. There are new vaccines being tested for all sorts of things. Cancer vaccines, Alzheimer's, vaccines to treat addictions, multiple sclerosis. All of these things are in various stages of development, which are really incredible. I think that maybe someday we will we'll have the technology where you can get a prescription for something. Maybe you're going through some cancer treatment, and the doctor can run some tests and email the data to the pharmacist, and a custom vaccine could be made for you, and the pharmacist could, not like a 3D printer, but a nucleotide printer, print it off and give you the injection there the next day. And I think that's where the technology is going, and it's just super incredible. It's kind of like Star Trek. It's very, very cool stuff. So this is the end of topic 20. I hope you enjoyed it. It is important for the midterm, and I will be asking you questions about general virus structure, general virus life cycles, and of course, coronaviruses and vaccines. If you have any questions, do come see me. I will see you in class.